The prokaryotic replication fork. What you need to know about DNA to understand replication is that DNA is a double-stranded helix, prokaryotic DNA is circular, DNA replication is semi-conservative, and DNA strands are antiparallel. Let's begin. Prokaryotic DNA has one origin of replication. This is the DNA location where replication will begin. The initiator protein binds to the origin sequence to unwind DNA strands. As the DNA strands unwind, supercoiling occurs in the DNA molecule. Gyrase is then attached to relieve the tension. DNA gyrase is a topoisomerase that relieves supercoiling ahead of the replication fork. DNA helicase then attaches to the replication fork and moves into the fork to break the hydrogen bonds and open up the DNA strand. Once the DNA is separated, single-stranded binding proteins bind to the single-stranded DNA to protect it, stabilize it, and prevent double-stranded DNA secondary structures such as hairpins. Now that the DNA fork is open, primates will produce an RNA primer to which DNA polymerase III will be able to attach and add nucleotides to the 3' end. This orientation is determined based on the sugar carbon of the nucleotide that is available for bonding. Remember how DNA is antiparallel? Well, this is where that comes into play. As you notice, one strand, the leading strand, is naturally oriented with a 3' end towards the replication fork, which means that the fork opens up, pole 3 can add nucleotides continuously to the strand. However, the other strand, the lagging strand, is oriented the opposite way, meaning that its 3' end is pointing away from the replication fork. This means that pole 3 will attach to the primer, then build a short strand of nucleotides called Okazaki fragment, and then it will reach the previous primer and stop. One of the DNA polymerase III molecule has now produced one continuous strand called the leading strand, and the other DNA polymerase III molecule has produced multiple Okazaki fragments, also called the lagging strand. Now there is RNA in our DNA strand. The RNA primers were great when we needed an available OH to attach to pole 3, but now we need to change those to DNA. That is when the DNA polymerase I comes into play. Pole 1 has proofreading ability, which means that it has 5' to 3' exonuclease activity, which enables it to remove the RNA primers, and since it also has 5' to 3' polymerase activity, it can add the missing DNA nucleotides. There are still some missing phosphodiester bonds in the strand, leaving nicks in the DNA. Ligase to the rescue. This protein will seal the nicks along to the new DNA strand. And this is how a prokaryote takes one circular double-stranded DNA molecule and makes an exact copy in a semi-conservative way.